Next presenter is Professor Timothy Hughes. Timothy has made key contributions towards the development and refinement of kinase inhibitor therapy for cancer. So we're sticking with the cancer theme. Through his pioneering work in chronic myeloid leukemia, he was a leader on many of the key clinical trials that underpinned the transformation of CML from a fatal disease to a treatable chronic disease, and pioneered the concept of treatment-free remission as a pathway to cure in CML. Please welcome Timothy Hughes.
Thanks very much. So my focus over the last 25 years has been chronic myeloid leukemia, and over that period I've been privileged to see it transform from a disease where it was virtually impossible to treat. Patient survival was 3 or 4 years on average, to a disease today where the 10-year survival is better than 90%. So it's been a very enjoyable journey, but I think also a journey where we've learned a lot of important lessons that can be applied to many other cancers. It all started with this remarkable finding from Noel and Hungerford, who discovered that every patient who had this chronic myeloid leukemia syndrome, where they had a very high white cell count, a very large spleen, they recognised for the first time, taking advantage of new techniques for identifying chromosomes, that every patient had this minute chromosome that was called the Philadelphia chromosome because they were working in Philadelphia. And this is the paper that they had in Science. That's the entire paper, not just the abstract. So very, very succinct. We should learn from that. And this is what they actually, eventually over the next 20 years, we learned what that identified, and that was this fusion between chromosome 22 and chromosome 9, which brought together the BCR gene with ABL, which was a tyrosine kinase gene. That then produced a chimeric messenger RNA and a chimeric protein, and it's that chimeric protein that drives the disease. But it's that chimeric messenger RNA that we take advantage of to measure in the blood, which gives us a very precise window into the dynamics of the disease, whether it's responding to therapy, whether resistance is developing, and that's been a critical part of the process of improving outcomes for patients with CML. It was 1990, so 30 years after discovery, that finally was proven that BCR-ABL, when inserted into a mouse stem cell and put into a mouse, caused a disease that was very similar to CML, really fulfilling Koch's postulate that this was driving the disease. And that then gave us a target. Can we actually target this overactive kinase, BCR-ABL, to actually develop an effective therapy? So this was BCR-ABL, and it relied on the binding of ATP to exchange a phosphate group to phosphorylate a tyrosine on a specific substrate, and once phosphorylated, that substrate then signaled downstream to drive proliferation of those leukemic stem cells, to prevent their apoptosis, and so it became clear that if we could inactivate the protein at this level, we would develop an effective therapy. And indeed, that's what was developed by Ciba Geigy, who became part of Novartis, when they developed a small molecule that bound very neatly into the ATP binding domain and competed with ATP for binding, therefore preventing or at least reducing the amount of phosphorylation of the substrates, and therefore either completely blocking or reducing this signal, driving the disease. And that was the first tyrosine kinase that was developed for a cancer. Since then, there have been over 30 different kinase inhibitors developed for various cancers, and I think the lessons we've learnt in CML have been important for many of the other developments that we've seen since then. So one of the things we did in the first few years when this drug became available was to develop a PCR assay along the lines of what Michelle was describing, with a log scale expressing the amount of leukemia present in the blood, which was a very close reflection of the total leukemia present in the bone marrow. And we were able to define a 3-log reduction in patients from the starting level as a major molecular response, and demonstrated some years ago that patients who could achieve that level of response had virtually no risk of progressing to a more aggressive leukemia and dying, which was the way that patients used to progress without effective treatment. And so that became the target of therapy for all of the future trials from that point on. But we also look at a deep molecular response to find as a 4-log reduction or deeper from the original level. Now, somewhat surprisingly, even though all these patients with CML have the same BCR-ABL protein and we use the same drug, we see highly heterogeneous responses, so that about 10% have what's called primary resistance. We don't see a significant fall in their level of leukemia. Many of those patients will go on to transplant, and many of them will actually progress. And other patients who have a good response and then have a rapid rise. In many of these patients, we were able to define there were mutations that prevented these kinase inhibitors from binding into the ATP binding domain and therefore allowing the kinase to be reactivated and for the disease to grow again. Unfortunately, it's from amongst these patients that we still see today about 10% of our CML patients dying of their disease. We have a small number who have a good response to get to a safe level, but not deeper for various reasons. And then patients, about 50%, have a fall that continues over time to the point where we cannot detect the leukemia anymore by our most sensitive assay available, RNA-based assay. So in about 2006, we had a number of patients in our clinic who by this assay were so-called PCR negative. We could not detect leukemia, and it became very relevant because these patients were struggling with the day-to-day toxicity of the kinase inhibitor drugs, whether in fact it was possible to stop and whether in fact we had eradicated the leukemia altogether. So we, with our first group together with a French group, to conduct a trial where we stopped the therapy and observed what happened thereafter. We monitored them very closely, and we found that after stopping, half the patients had a very rapid rise. They were heading back towards complete uncontrolled leukemia, went back on their drug and went rapidly back to a deep response. But the other half remained PCR negative. In fact, to this day, I have patients who went into that trial in 2006 and are still PCR negative, off therapy, and I think they're cured. But that's an open argument. But the critical point was there's a real dichotomy, either dramatic rise or ongoing PCR negativity, suggesting that if we could understand that process, we could greatly expand the availability of this very desirable outcome. Because at the moment, only 25% of our patients overall can achieve this optimal outcome, and the majority of patients are still on their drug or struggling with resistance or in fact have died of their disease. So we have a long way to go in CML, even though our survival now is so good at 5 and 10 years. So this was the way things looked for our initial trial of treatment-free remission, where we stopped the drug. We saw a lot of patients lose their response and go back on drug, but then we saw this very impressive plateau, and we asked the question whether it was possible that these patients had in fact eradicated their leukemia altogether. So to answer that question, we developed an assay very much along the lines of the assay that Michelle described, which was another log more sensitive, which was a DNA-based assay, patient-specific, to interrogate whether they still had leukemia. And in fact, we found that all of these patients who were still PCR negative by the conventional assay had positivity by this much more sensitive DNA-based assay, some patients on every occasion, some patients on some occasions, suggesting that we hadn't actually eradicated the leukemia altogether, but had merely brought it down to a level where it wasn't reoccurring, either because it was incapable of regrowing or because there was an active immune control of that process. And that's what we're still trying to understand. We did go on to look at these patients specifically, asking the question, well, if they have positive leukemia and it's not regrowing, what is it? What are these cells that are positive in the blood? So we flow sorted the blood and separated into the different components, the B lymphocytes, T lymphocytes, all the different cells that are in the blood, and we found that there was a lot of positive leukemia present in B lymphocytes and T lymphocytes, but none at all in the granulocytes. And this is very interesting because these are, in many cases, long-lived cells that don't require a stem cell to produce them in the recent past, whereas these cells require an actively proliferative stem cell to produce granulocytes. So it told us something about the cells that were surviving. We then asked the question whether, in fact, we could predict who would relapse and who would remain in TFR by characterizing the granulocytes at the time of stopping. And in fact, we've since shown that patients who had positivity in their granulocytes all relapsed when they stopped, and none of the patients who were successful had positivity in their granulocytes at the time of stopping. And this now has become a test that we are attempting to widely disseminate, which is a so-called red light, which you would identify patients who should not stop and should stay on therapy or look at an alternative approach. We've now got on to do a number of very detailed immune studies, which I don't have time to expand on, the work of Agnes Yong and Yaz Irani, demonstrating that the immune system is critically important, and it's the balance between the immune effectors and the immune suppressors that seems to be very, very predictive of who can achieve treatment-free remission. So it's not just a matter of reducing the leukemia by tyrosine kinase inhibitor effects, but you also need to have understanding of the immune-mediated leukemia control, and that's where we're really focusing today by looking at better ways to deplete the critical leukemic cells, but also how to modulate the immune response. I'd like to finish there by acknowledging the many collaborators in Adelaide and around Australia, critically the patients who have contributed, some of them over 20 years or more, to the clinical studies that we've conducted, to acknowledge the support of the various funders there, and to thank particularly my family and my wife, Lyndall, who's in the audience, who's been a great supporter over many years. Thank you very much.
Thanks very much, Timothy. So I open this presentation to the floor for questions. Yes, we've got one at the back.
Lovely talk. You mentioned the hoping that the immune system might be able to control or is controlling some of the cells. The dogma is that it's the number of mutations that are in the tumour cells that gives the immune system something to grab onto. So how many mutations are in, other mutations other than BCR-ABL are in the leukemia cells, and are there any particularly interesting ones?
Yeah, that's an area that we weren't able to really expand on. I wasn't able to expand on today, but particularly in those patients that we see a poor response initially, we've identified that there are additional mutations besides BCR-ABL, P53, ASXL1, that seem to be driving other pathways, and therefore simply using a kinase inhibitor is not going to be effective. We see that in about 30% of our de novo patients. The other 70% seem to be solely driven by BCR-ABL, and that's why we get such dramatic responses in many patients. It's very hard to characterise the mutations down the track when we have very, very low numbers of cells, but that's certainly the situation at the start, and we see more mutations in the patients who develop that acquired resistance.
Francis?
It's a very general question. Are there side effects of taking these therapies for 5-10 years?
Absolutely, yeah. I didn't have time to talk about that, but that's what really drives us to try and towards treatment-free remission. There's a substantial increase in the risk of vascular toxicity, heart attacks, strokes, etc., that our patients have to face. Day-to-day toxicities, GI toxicities. So the quality of life is actually substantially impaired for many, many patients. So it's a real challenge. They know they're not going to die of their disease, but they have to put up with the drug, which has quite unpleasant side effects. So that's the driver for treatment-free remission.
Timothy, thanks very much indeed.